But I'd like to start out by drawing your attention to the nucleosome core, which, as you can see, is a tight left-handed spiral of histones that's tightly wrapped by DNA. And this creates impediments for active processes, dynamic processes such as replication, transcription, repair, transcription factor binding, whatever, has to deal with a landscape that's dominated by nucleosomes. And we've been keen to understand how this can occur. However, we've found over the years that the methods that have been available for taking a genome-wide method, a genomics approach to understanding these dynamic, this dynamic situation have really not been suitable for the task. So this led us to develop some of our own, some of which you just heard about. But what I'd like to do is start out and telling you about the methods, how they work for mapping the chromatin landscape, and then and where we are right now in developing these methods. But then turn to some interesting questions in chromatin biology. The first one of which is the dynamics of chromatin accessibility, which you might not have thought was actually dynamic. And then what is driving that, chromatin remodeling dynamics. And then a new direction for us, which is RNA regulation of chromatin. And I think you'll see at the end that actually there's an interesting connection between these three topics. Okay, but let's start out with the mapping question. So you're certainly probably familiar with chromatin immunoprecipitation. It's been around since the mid-1980s, and ChIP-seq has been the go-to method for chromatin profiling for the past 15 or so years. However, it's not the only way you can profile chromatin. There are methods that are referred to as enzyme tethering methods, an early one of which was introduced by Bas van Steensel, what we call DMID, where we use a fusion protein between E. coli DAM methylase and a chromatin protein of interest. DAM will then methylate the adenine of GATC sites in its vicinity, and then it's a matter of mapping where the methyls are. Then a few years later, Uli Lemley and his group introduced chromatin endogenous cleavage by substituting micrococcal nuclease, or MNase, for DAM, and this takes advantage of the base pair resolution that's possible with MNase. And when Gabe Zentner was a postdoc in the lab, he converted this from a Southern blot readout into a full genome-wide sequencing-based method. However, it's this other method that appeared in the same paper from Lemley's group called chromatin immunocleavage that we've done the most with. And our version of it goes like this. We mix live cells with lectin-coated magnetic beads, they stick. We then permeabilize. We then add an antibody, say to a transcription factor, follow that with a fusion protein between protein A and MNase, or protein A will bind to the antibody. We'll tether the MNase, which we can activate with calcium. And in our version of this that we refer to as cleavage under targets and release using nuclease, or cut-and-run, we allow the complex to diffuse out into the supernate. We extract the DNA, ligate on adapters, amplify libraries for sequencing. When Hatice Kayakur was a postdoc in the lab, she substituted TN5 for MNase, where TN5 is the cut and paste transposase. That's the basis for the Illumina and Xtera system and the ATAC-seq method. But in our case, we're tethering. And this we then activate with magnesium. The TN5 drops its payload of DNA sequencing adapters. We then extract the DNA, amplified libraries for sequencing. And this goes very fast. It just takes 1 day from live cells or nuclei to sequencing ready libraries. Now, one nice thing about this, in addition to being fast, is that the backgrounds are very low. And that's because, unlike ChIP, where you cross-link and solubilize the entire cellular contents, and then add an antibody to pull a needle out of the haystack, with both of these methods, we're only solubilizing the little bit of DNA that's in the vicinity of the antibody, and that keeps the backgrounds low. Now, we've been trying to improve cut and tag by getting rid of this DNA extraction step. And we can do that by adding a little bit of SDS and heating it up. Now, what that does is the TN5 after tag mutation is very tightly bound to the DNA. And so the SDS will release that, but then because that can inhibit the PCR, we add an excess of non-ionic detergent triton, and then we can carry out all the reactions in a single PCR tube. Now, as we're finishing up this CUT&Tag direct method, the pandemic hit, I had to shut down the lab temporarily, and then I realized, oh, you know, this is pretty easy to do. Why don't I just bring it home and set it up in the corner of a bench in the laundry room in my house. And this is really all you need to do it. And as I'll show you, you get high quality data, pretty much surplus material that we don't use in the lab, still doing it now. And the data that we get is really quite nice, whether we do it at home or in the lab or by the extraction method. It really has this advantage over ChIP-seq in that with ChIP-seq, because you're sampling from the entire genome, you basically are going to get a low background, and then it's a matter of building up the features over that background. So in this case, you're looking at H3K27 trimethylation human K562 cells. And I'll be talking a lot about this histone mark. It's a mark of polycomb silencing. And as you can see, it gives broad domains on the order of hundreds of kilobases wide. And what you can see is that because you need to fill up the background, you're going to need a lot of unique reads. And so you're going to need a lot of cells, about 1 million cells typical. We can go down here to about 100 cells or so and get good feature definitions. So it's orders of magnitude more sensitive. And we get pretty much the same result, whether it's in the laundry room or in the lab. Now, because of the simplicity of the method and the high data quality, Derek Jansen's postdoc in the lab has automated this, and AutoCut&Tag follows from AutoCut&Run, which is a mainstay in our shared resource facility. It's done thousands of samples for my colleagues at the Fred Hutch and the University of Washington. But what we see here is that we can apply this AutoCut&Tag conveniently to patient samples, and it's fully automated. Now, this just shows an example of a particular patient sample. I'll say more about it later, but what you're looking at here is we can use different histone marks in order to get to identify different parts of basically different readouts from each of these histone modifications. And Derek has applied this to mixed lineage rearranged leukemias. Now, these are leukemias that are driven by fusions involving the KMT2A gene, where KMT2A is this enormous protein with a set domain at the C terminus. That set domain will methylate histone 3 on lysine 4, but it's the N terminus DNA interaction domain that's involved in the fusions. And after a breakpoint region, there can be a histone interaction domain, typically from these 4 chromatin regulators, but some 80 different fusion partners for KMT2A have been identified. And to make matters even more complicated, sometimes they present as lymphoid, sometimes as myeloid. Sometimes they'll present as lymphoid, then after treatment, come back as myeloid, so-called mixed lineage leukemias. And we were wondering whether there's an epigenetic component to this that might be responsible for this kind of switch. To get at this, Derek used 2 different antibodies to identify oncofusion protein targets. One to the N terminus that picks up both the wild type and the oncofusion protein, and the other to the C terminus that only gets the wild type. And here in a well-studied cell line and looking at 2 known targets in the cell line for KMT2A fusion protein, what we see is that when they're normalized the same, we see a much stronger signal at the N terminus. We're picking up the oncofusion protein as well as the endogenous over the C terminus in both cases. Now, this was done with CUT&Run, and then using CUT&Tag profiling, Derek noticed something interesting for H3K4 trimethylation. This is a mark of active promoters, and the promoters are indicated by these arrowheads here. But what you see is that we're getting aberrant methylation in the gene bodies here in particular places, just where the oncofusion protein is bound. And this suggests to us that maybe we could use this mark as a proxy for following the course of treatment in the clinic. And as a proof of concept here, we tried this out on 2 inhibitors that are used in the clinic, a .1L and menin inhibitor. And although we didn't see much effect for the .1L inhibitor, we see that there's strong suppression of the aberrant methylation for the menin inhibitor. And this is done at doses that are well tolerated and in a proliferation assay for that are quite effective. So we think that this can be used in the automated format for following patient samples in the clinic. Now, what I've shown you so far are just some methods that we developed back up until a few years ago. But in the meantime, CUT&Tag has really kind of taken off and for both single cell and spatial profiling. Just list here the methods that have been developed, mostly by others that take advantage of the features of CUT&Tag. So one is there's a couple of papers using different readouts from Xiaowei Zheng and from Rong Fan to do spatial CUT&Tag kind of profiling in different ways. But what's become quite popular is to use it for single cell profiling. And that's because you carry out all the reactions in a single tube, and then you can use high throughput dispensers in order to get lots of cells for single cell profiling. I'm not going to go through. I'm going to go through just a couple of them. These are ones from our lab that I'll just touch on briefly, and then but mostly I'm going to be talking about what we've done in order to study some interesting problems in chromatin biology with the first methods. Okay. Single cell profiling, here's an example that is actually built on the study that I just told you about from Derek Jansen's about these KMT2A fusion leukemias. Now, when Derek profiled these two marks, the ones I've already talked about, one for active promoters, K4ME3, and for polycomb silencing, K27ME3, typically they're mutually exclusive at promoters, but often they're bivalent, as you can see here. This is a concept introduced by Brad Bernstein and others many years ago. It's been a bit mysterious, just what it's about, where you get both. You get both of these marks at the same place. And I'll say a lot more about bivalency later, but for purposes of the methodology, what Derek noticed is that there's inconsistent oncofusion protein targets when you go from one tumor sample to another, leukemia samples. You see differences in where KMT2A is bound. So what's going on there? In order to sort this out, he took a single cell approach. So this shows clusters in UMAP space of 8 different histone. Of 8 different primary leukemias or cell lines, and we get, in both cases, we get 8 different clusters. Okay, that's fine. But let's drill in on this one cluster here for this primary mixed lineage leukemia. Now, if we look at target loci that are targets in various different leukemias, these are driver mutations, these are driver transcription factors. For example, if we look at the H3K27 trimethylation at HOXA9, we see a tremendous amount of variation from the same tumor in the amount of H3K27 trimethylation. So and we see it's quite different at the different loci in the same leukemia. So this suggests to us that maybe there is some kind of epigenetic instability that's going on here that might be responsible for the kind of switch, aberrant switch that occurs in the mixed lineage leukemias. And this is an idea that the lineage plasticity is really somehow tied in, particularly with H3K27 trimethylation loss. And this is something that Derek is currently following up. But it'd be really nice if you could look at both marks in the same cell, and we can do that thanks to the work of Mike Mears, a postdoc. He did this as a postdoc in the lab. Mike has just started his own lab at Washington University in St. Louis. But let me just explain this particular strategy. And that is, you conjugate one of the 2 adapters that's needed for amplification after tagmentation to a primary antibody, and then carry out the tagmentation reaction, you take a second antibody, a second set of conjugated primers, conjugated adapters, and you repeat it multiple times. Then follow with the other adapter that's needed for the amplification and carry out the tagmentation reaction there. And this will give you a multifactorial readout. Now, the application is actually described in the paper. I won't go into it, but I'd like to show you how we can get a lot of cells at low cost in order to do this. You need a lot of cells for single cell profiling. And you can get that here by using a combinatorial indexing approach. So this is just a proof of concept of it. We take two different, very different cell types, and we mix them and then carry out the bulk multi-tag for getting in the barcoded adapters in each different adapter sets in each of the wells of 96 well plate, then mix them and array them in 96 well in 5,184 well format. That's 72 by 72 array on a chip in about 10 cells per well. And this is very inexpensive relative to what you'd have to do in order to get a high throughput readout. And we get about 50,000 cells in this way. And just to show you this actually works. So here we see the separation of the two cell types, not surprising, using either the marks or in the joint projection. Just point out that even where there are two cells that happen to come together of different types, they still end up falling within the same clusters. So, and we've since come up with ways of actually cleanly separating those guys out. So I think this is a nice way to do single cell profiling. It's relatively inexpensive because you can get a lot on a single chip. Okay, that's what I wanted to say, at least at the outset, on the methodology. Now let's turn to some interesting questions. So first of all, chromatin accessibility dynamics. Now you might say, well, no, what? Is chromatin accessibility dynamic? I thought it was just open. Well, open chromatin is indeed the term that's used to describe accessible sites. And that's based on this kind of model where you have enhancers that are bound by transcription factors and promoters that are bound also by RNA polymerase II. And this is open and everything else is closed because it's covered with nucleosomes, right? So it's open and closed. Where's the dynamics? Well, actually, this concept of open chromatin goes way back. I mean, it was really introduced back in 1980 with DNase hypersensitivity mapping of promoters, for example, in these two papers. And ever since, this concept of open chromatin has been continued using different kinds of probes. So DNase-1 was the first one, and then you have MNase and restriction endonuclease, DNA methyltransferase, transposase, nicases, and also you can get accessibility mapping just by cross-linking and sonicating, and where the chromatin breaks most readily turns out to correspond to accessible sites. So just doing that's the input for chip, and actually you can get accessibility mapping out of it. And what this emphasizes the fact that each of these methods is a method unto itself. If you get a difference, say, between DNase-1 hypersensitivity mapping and AICC, I mean, who's right? There's no ground truth validation. So that's a problem with this. And it's one that, well, we didn't set out to address, but it turned out we sort of stumbled over an answer to this. And this comes from when we developed CUT&Tag. There are two parts to the fusion protein. The protein A part is bound to the antibody. So that does the targeting. But the TN5 part is a bit of an annoyance for us because it will bind very tightly, as I pointed out, to accessible sites. And that's an artifact for us because we won't be able to tell whether it's targeted or accessibility. However, we found that we could suppress this artifact using 300 millimolar sodium chloride. And the salt competes for the electrostatic interactions between the TN5 and the DNA. So that suppresses the artifact, and this works really well. So here we demonstrate this by using H3K4 dimethylation. It's a mark for both enhancers and promoters, and so is AICC. So we line it up over AICC from the ENCODE project, the community standard here, for K562 cells. And what we see is that there's nice signal out about, on average, about a kilobase on either side of the accessible site, with very little, if any, tagmentation in the accessible site itself. So this works pretty well. And this goes for all TAG-seq sites basically. But we found that if we leave out the salt during the tagmentation, it's as if all of the TN5 gets sucked into the hole in between to give us what we call cleavage under target accessible chromatin, or CUT&Tag, which is a chromatin accessibility method here. And our interpretation of this is that if the TN5 is anchored to nucleosomes in the vicinity, there's enough flexibility that if there's a large enough gap in the chromatin landscape to fit two, with enough DNA in between that we can sequence it, then you'll get this signal where you won't get it in linker regions on either side. And this predicts that mainly this signal would be driven by small fragments, and indeed it is. If we look at less than 120 base pair fragments for the CUT&Tag signal, we see that's where most of the signal is. Now, it actually corresponds to all the ATAC-seq sites that we can map. So this is taking one of the two replicates from the ENCODE ATAC-seq data to use to call the peaks, and the other replicate to line up over the peaks. And you can see that we basically get everything that you get with ATAC-seq. Then we began to wonder about RNA pause PAL2. Now, I'll be saying a lot more about this later, but for purposes of discussion right now, pause PAL2 is stuck in between in the accessible site, just up against the first nucleosome on either side of the gap. So we wondered whether we could tether to pause PAL2. We can do that using a mark to the heptapeptide repeat that is present 52 copies on the C-terminal domain of the largest subunit of RNA PAL2. This is serine 5 of the heptapeptide repeat, so it gives us good signal. And it turns out that this works actually a little bit better than, because it's stuck in the hole here, it works a little bit better than the histone mark. And if we look closely here, what we see is that the small fragments actually correspond to the ATAC-seq sites. If we just look at small fragments, the difference is with ATAC-seq, you're also getting insertions in the linker region, so you get this broader region. But what I've shown you here is, since pause PAL2, I mean, it's been around since 2 billion years, so actually you couldn't have anything better that to give you ground truth validation for accessibility methods, not just for CUT&Tag, but also for the other accessibility methods. This is what they're proxies for. This is what you're looking at. You're looking at, in both enhancers and promoters, you're looking at what's happening in the wake of pause PAL2. And I'll be saying a lot more about that through the rest of the talk. One thing that we like about this is that we get nice, not only nice resolution, we get, if we downsample and compare to ATAC-seq from the ENCODE project, we get more peaks and we get a higher fraction of reads in peaks, so it gives us high data quality. And because of the high data quality, we came up with what we call CUT&Tag 2 for the price of 1, to get both the repressive regulome, which is H3K27 trimethylation, and the active regulome, that is pause PAL2, in the same experiment. So we're just doing exactly the same thing that we do with ordinary CUT&Tag direct, but in this case, we're just mixing the two antibodies at the beginning, and then coming up with a Bayesian deconvolution algorithm, 2-for-1 separator, developed by Dominic Otto, who's a postdoc with Minusetti here at the Fred Hutch. And the basic idea of the deconvolution is that you've got two kinds of clues that you can use to update when you go through the algorithm to basically update your priors. And that is, one is that you've got small fragments for the PAL2 serine 5 phosphate and large fragments for the nucleosomes, and you can separate those out. And the other is that you have this large domain, as I showed for at the beginning, broad domains for K27 trimethylation, but of course you get sharp peaks for the accessible sites. So all of that allows for a clean separation with the same workflow. So that's what I wanted to say about chromatin accessibility dynamics, and to show you that RNA pol2, PAWS pol2 plays a central role in chromatin accessibility. But now let's turn to the more mechanistic question of what is actually driving this. And this gets us into chromatin remodeling dynamics. Now, the question is, how are accessible sites targeted for histone eviction to get the whole thing started? And we know a lot about that in yeast, and this is studying the RISC chromatin remodeling complex. It's a multi-subunit complex. It's the only essential remodeler in yeast, and its job is to keep yeast promoters free of nucleosomes. So that's what it does. It does it by targeting promoter motifs. And these are targeted by a DNA interaction module that has 2 DNA binding proteins in there. So it's directly targeted. Now RISC has orthologs in animals called the BAF, Brahma-associated factor complexes, where you can see from the color coding that most of the subunits are really shared between the two. For example, SDH1 is the catalytic subunit of the RISC complex, whereas BRG1 or BRM are subunits of the BAF complexes. But what's missing from the structures is that there's no DNA interaction module that's inherent to the structure in the BAF complex. And so this raises the question, how does BAF know where to go? And of course, from what I've already showed you, a good candidate here would be PAWS RNA pol2. Maybe that's what is bringing it in some way. So, and also, we're quite interested in BAF because it's associated, BAF is mutated in more than 20% of human cancers, as are many other people. So there's been a lot of studies of BAF. But Sanpan Brahma, postdoc in the lab, decided to take a small molecule approach in order to address this question of whether RNA pol2 is involved here. And to do this, he used inhibitors. One inhibitor, triptylide, inhibits the opening of the pre-initiation complex by RNA pol2. This is a helicase that it inhibits, so the DNA can't denature to push the denaturation bubble forward. Or actinomycin D, it acts as a roadblock. It intercalates in the DNA and it prevents PAWS release just physically, but we can also inhibit with flavopyridol. Flavopyridol inhibits the CDK9 kinase, which phosphorylates serine 2 on this heptapeptide repeat that I mentioned earlier. And by inhibiting PAWS release, you don't get removal of serine 5 phosphorylation, and you don't get addition of any more serine 2. So we can use serine 5 phosphorylation to tell us what's going on at the PAWS site. And that is it with triptylide, the PAWS pol2 will disappear. It'll get chewed up because it can't go anywhere. And with actinomycin D or flavopyridol, it'll build up at the PAWS site. So that's what we're going to look at. The same mark that I mentioned just earlier, we're going to do CUT&Tag on here. And here, this is done in mouse embryonic stem cells. And when we add triptylide, as expected, you see that pol2 gradually disappears. Now, if we look at the BAF complex using an antibody to BRG1, the catalytic subunit, we get a nice BRG1 signal. And we see it also goes away, though more slowly, which implies that RNA pol2 is somehow stabilizing BAF at its site. Now, we also can use flavopyridol, which inhibits PAWS release, and we very quickly get buildup of the PAWS pol2. And we see the same thing going on with the BAF complex, which also confirms that it stabilized BAF on chromatin. And we see basically the same result. even stronger with actinomycin D. And so this implies that PAL2 is targeting BAF for promoter clearance. Now, we wanted to get into the mechanism of what's going on here. And in this case, Sandepen applied a method that he developed previously for studying the RISC complex in yeast. And that is to follow CUT&Run with CHIP to see what actually is going on in the intermediate that is in the BAF intermediate in this case. So using an antibody BRG1, we carry out CUT&Tag in the usual way, but then we take the CUT&Run the usual way. Then we take the supernatant and use it as input for CHIP. We first get rid of the BRG1 antibody with a peptide, and then follow that with the secondary antibody, and then carry out the second antibody, and then carry out a CHIP in the usual way. Now, the advantage of this is that we can see, if we use a histone modification mark, we can see if there's a nucleosome associated with it, and with the complex, first of all, and we can see whether it's fully wrapped or would look like an intermediate that is partially unwrapped, because we know that BAF can unwrap nucleosomes. Okay, so he applied this in mouse embryonic stem cells, and looking at foci for NANOG, nanos of pluripotency factor. This just shows the genome. This is if you line up all the NANOG sites throughout the genome, the foci where NANOG binds. And what we see is that we get nucleosome-sized fragments, and we also get smaller fragments. Now, some of these will be NANOG and other transcription factors. However, we can distinguish whether these small fragments are actually nucleosomes by using CHIP, from taking the supernatant and using CHIP, in this case, for mark of enhancers and for mark of promoters. In both cases, we see that there's histones in there. So this says that what we're looking at here are partially unwrapped nucleosomes that account for these small fragments. Now, we then add flavopyridol, and of course, that will, as I showed you, that will cause a buildup of more of the RNA PAL2 and more BAF. And what we see is that there's almost complete eviction of the nucleosome. So we can map the dynamics at these sites. Now, we can also do this in a different way. We can upregulate NANOG using a change in the culture medium. That's commonly done. And we basically see the same effect. You get increases overall, but you actually lose, you get you get this, again, this loss of the nucleosome at the accessible site. And we can confirm that genome-wide, it's not just in NANOG sites, but everywhere, we're basically seeing under these conditions, we're seeing basic increase in accessibility overall. And we can also look at the silent chromatin, and we see that with silent chromatin, you see the same kind of effect. And this fits with other work from other people showing that BAF remodeling opposes polycomb silencing. Now, let me turn to this question of bivalency that I introduced earlier. And what we can see is that this just shows the bivalency that has been was shown many years ago in mouse embryonic stem cells, where you see these two marks that are co-occupy. And then if we look at RNA PAL2, we see that there's a signal, and the signal greatly increases with either of these drugs that causes increases in RNA PAL2 and increases, cause PAL2 and increases in BAF. We also see that, and we see the same thing with BAF here. So basically the same kind of process is going on in bivalent chromatin as is going on at these sites, at these accessible sites, but it's over a broad region here. And this leads to this kind of model where the real difference is having a transcription factor present in high enough concentrations that it can get in there and bind and be involved in this cycle of BAF and RNA PAL2 working together and the transcription making room for the transcription factor, and transcription factor comes in and will complete the cycle. That is where there's good evidence, at least RNA pol2 undergoes abortive initiations over a second time scale. I like to think of this as a cycle, or this is the way we're thinking of it, and not as recruitment in any way, because these don't necessarily touch. They're working independently, just like in a combustion chamber where you have valves, spark plugs, and pistons working independently, but in a cycle. So I'd like to leave you with this idea that this is really what's going on at these accessible sites. You have this independent action of these three components that, but requires a transcription factor in order to provide the specificity. Okay, finally, I'd like to turn to this RNA regulation of chromatin. And the motivation for this is that we know of chromatin regulators, exist as the best studied, the best understood, exist, coats the inactive X chromosome in mammalian females. So here you see that exist is expressed from its locus, and it coats and cysts the inactive X, where you don't see that over the active X. Now, the methods that have been used for studying cysts and other long non-coding RNAs are, and other kinds of processes such as RNA modifications, is immunoprecipitation. So RNA immunoprecipitation is embodied in all these different methods here that are described here for different purposes. And the problem is that you really need a lot of material here. You need a lot of cells, and you have to sequence pretty deeply in order to do this. And we wondered, well, maybe we could find a cut-and-tag solution for getting better efficiency for this particular process. And this is work of Nadia Kaja, postdoc in the lab, together with Kami Ahmed, my collaborator at the Fred Hutch. And this is just cut-and-tag workflow, but it's applied to proximity labeling of chromatin-associated RNAs. It goes like this. It's the same workflow, begins the same way. We bind to beads, bind a primary antibody. Then we bind a streptavidin conjugated secondary antibody. And that allows us to attach an oligo DT primer connected to one of the two adapters that's needed to do the amplification after tagmentation. Now, we then add the TN5 together with the other adapter. So everybody's in the same place now. And then we add reverse transcriptase and with the magnesium to activate to get simultaneous reverse transcription tagmentation. So the oligo DT will prime the poly A, and as reverse transcriptase goes along, when it gets close enough to the TN5, the TN5 will use it as a substrate because it will tagment, as others have shown, it'll tagment RNA DNA hybrids. And this puts both adapter sequences on there so we could do the PCR and sequencing with the same workflow. And one nice thing about this is that we can apply this to different situations. We can look at RNA protein interactions, RNA chromatin interactions, RNA modification interactions, and it's complementary to these other methods in that we're doing proximity labeling. We're not doing direct binding. And let me just, I'm going to illustrate this with the examples where we first applied it. And we applied this all in Drosophila, but we've also got it working now in mammalian cells. So ROX2 is a long non-coding RNA that is stabilized by the dosage compensation complex in Drosophila, which coats the Drosophila male X chromosome for twofold upregulation. So it's a form of dosage compensation that's totally different from what I was showing with exist. And when Nadia uses an MSL2 antibody, where MSL2 is part of the dosage compensation complex and binds to ROX2, so we use this antibody and compare it to IgG, we get lots of positives where ROX2 turns out to be the winner overall. But what about these other positives? And it turns out that if we look over the chromosomes where they map, this is the Drosophila karyotype. And what we see is that, that we see these positives over all the chromosomes, but especially high on the X chromosome. So what we think is happening here is that because of proximity, because we're looking at an antibody that's bound over the entire X chromosome, it's trapping and it's priming off of the nascent transcripts that are being expressed. So we think that this is sort of background labeling, but it does sort of prove that actually what we're looking at is successful RT and tag. Okay, so that's proof of concept. And now we apply it to H3K27 trimethylation, which is the mark of polycomb silencing. As I mentioned, we get a lot of positives. We get some uncharacterized long non-coding RNAs. We don't know what they do. Point I want to make here is if we do cut and tag for H3K27 trimethylation, we see that's where the positives primarily go relative to the non-enriched. And what we also see is that if we look at active marks, it's strongly depleted. And this implies that what we're looking at here are very low level transcripts that we're trapping here on the chromosome. So this shows that we can also use this for modifications for RNA. But let me turn to the use for looking at RNA modifications, because there's an antibody there, we can use the same trick. And this seems to work pretty well. So what we see are some positives, and this just indicates that some are actually known to be long non-coding RNAs that are present there, and these are known to be depleted. And if we look at the enzyme metal-3, which accomplishes, which does the methylation, we see that metal-3 by cut and tag is for these enriched, but not for the depleted, is primarily located over the transcriptional start site. And so we took a look at our, and then the knockdown just shows that you lose the signal when you knock down metal-3. And we looked at RNA pol2, and what we see is that with cut and tag, these enriched MEM6A enriched sites are actually primarily located over the transcriptional start site. That's not what we see for the depleted or the non-enriched. So what this implies is that paused RNA pol2, which is quite abundant here, as I've shown you, might be a landing pad for metal-3 in order to accomplish this. So it kind of brings together the three parts of my story here, that high pol2 pausing predicts high M6A. We don't know any mechanistic consequences of this, but I think it's a place to start. So to summarize, I've shown you that we can do chromatin profiling using cut-and-run, cut-and-tag, attack, RT and tag. We can do multifactorial, multi-tag, 2-for-1. Take-home lessons are pause pol2 defines accessible chromatin, stabilizes BAF, and predicts M6A enrichment. And I'd just like to acknowledge people whose work I talked about, particularly Sandapan Brahma, who did the BAF study, Derek Janssens, who did the leukemia study, Mike Mears, who developed multi-tag and many other things when he was in the lab, and Nadia Kaja, and Kami Ahmed for the RT and tag study. And if you want to try any of this, just go to protocols.io, and we have a question and answers that will, if you run into any problems, we'll do some troubleshooting for you. This is the lab, this is where we work, and here are our funders, and thank you for your attention.
That's great, thanks, Stephen. A real combination of different techniques there. So we have a pile of questions here. Many of them are sort of a little bit technical, but I think they're at least about the applications of the method. So I wondered, can you see the question list there, or
oh yeah,
the Q&A box?
Oh yeah, oh wow, a lot of questions. Do you alter the Charlotte Barkley? Did you alter the protocol at all when you use CUT&Tag for low cell numbers? No, actually, we just use the same protocol for everything. Aaron Mahesh, both CUT&Run and CUT&Tag work well with histone modifications for transcription factor profiling. Which method do you think is best? We strongly prefer CUT&Run for transcription factors. And that's because for exactly the same reason I pointed out, that we suppress the ataxic artifact by adding salt. It will also destabilize transcription factors. Anything that's not tightly bound, it doesn't have to be directly bound, but it has to be tightly bound. Nucleosomes, the DNA wraps around, so they hold on. But transcription factors will get destabilized. We can do some of them like CTCF, which is strongly bound, but I wouldn't recommend it. CUT&Tag works really well for that. It gives you better resolution overall. Joshua, okay, I won't give the names, there are too many questions. We often use the background in ChIP-seq to normalize the signal extraction saline, and view the signal as a signal noise ratio. In the absence of spiking controls, how would you recommend normalizing CUT&Run and CUT&Tag data when the background is low? Well, let me just point out that I pointed out that when you crosslink and sonicate, you can use that as an accessibility method. And when you use the input for ChIP, what you're doing is you're normalizing out that accessibility artifact. Okay, we haven't found that to be really necessary, and that's because of the high quality data we get without that. So we'd like spiking controls, but we don't actually, we just use that as a global calibration. We don't do any normalization. That's for ChIP. It's not for CUT&Run or CUT&Tag. Can you adapt? Oh, can you adapt CUT&Tag to formaldehyde crosslink cells? I need to fix my material before processing to CUT&Tag. You really don't have to do that. I just recommend trying it. You don't have to crosslink. I think this is a misconception, but we found that actually when you're using a nuclei, it's a good idea to do a very light cross-linking, because more cross-linking, the more you're getting epitope masking. Your antibody might not work, so I wouldn't recommend doing it. Just try it. It'll be okay. It's a misconception that you have to lock things down. Under physiological salt conditions, you really don't, everything should hold together. After all, it holds together in the cell. We're only permeabilizing and the nucleus stays intact. So everything should be there. Okay. Trust me on that. Can CUT&Tag be used to identify chromatin remodeler localization? Is it better to use CUT&Run? It's a good question. I did show you CUT&Run, which is where we've, okay, we've used both. I showed you both CUT&Run and CUT&Tag. I don't know for the risks, the BAF study, which by the way is up on bioRxiv, so you can take a look at it and maybe you can do the comparison. The work I showed you for the Sandeep and Brahma. It's out there. We often see papers in study where CUT&Tag is used for epigenetic mark, much less for, okay, I think I already answered that. Yeah, use CUT&Run for transcription factors. If you target active chromatin in CUT&Tag and CUT&Tag 2-for-1 with an antibody for RNA PAL2, don't you risk for promoters, gene bodies, and enhancers? No, actually, that's a misconception. This has been shown by John Liss and Charles Danko that actually enhancers also have PAL2. They just have much less of it. And that is, it's the same chromatin context for both. However, you just have more stuff going on at promoters than enhancers. Yeah, so what I was showing you was for both promoters and enhancers. When I lined it up over TAC-seq sites, you could see that. Can you decipher bivalently marked promoters in the 2-for-1 Bayesian convolution? Yeah, oh yeah, we do that. That's routine. Yeah, that works for that. One paused RNA PAL2 underestimate chromatin accessibility. I don't know why you would think that. What about other DNA templated processes such as repair that also need open chromatin architecture? That's a good question, and it's not something we've looked at, but we would like to look at that. Or you could look at it yourself. All the methods are out there if you want to try it. Does not CDK9 regulate BAF? I don't know. Maybe it's something we should look into. Thanks for your question. Okay. Am I going to get through all of these or? Because I see that there's some panelist questions here. Maybe I should take one of those and hold off on the others.
Yeah,
Yeah.
absolutely, Stephen.
Okay, was there a panelist question or?
Vijay, I guess you had your
Yeah.
hand raised.
I was waiting. I mean, Stephen was breathless reading all the questions and answers. So, okay. So, I mean, I was very intrigued by the RNA chromatin profiling you could do. So this is excellent advancement because this was technically so challenging for years. So thanks a lot for that, actually. This is amazing. And
yeah. Yeah. And actually, we've been looking at EXIST and other chromatin and other long non-coding RNAs. I didn't talk about this, but it's looking like it works really well. We're using a female cell line for EXIST. I can't remember which one, but I think it'll generalize. And we're following it up with other things. For example, one thing that we're trying to do, and I think we've got it working pretty well, is to make it so you're not limited to the 3' end as you are with this, because you're doing the oligo. So you're not dependent on oligo DT. That's a little trickier because you have a lot of contamination with our DNA, but we've got that working now. So I'm quite keen to expand the use of RT and TAG.
on the last slide, you showed that when you have paused polymerase or a longer pause causes higher levels of M6A. Is that what you showed, right? If you have a longer polymerase pausing, right?
Yes, right. So at the transcription start site, so our idea is that this is a landing pad. Like making this accessible is also good for M6A to sit, for metal 3. It's a part of a complex to sit down and to get access to its substrate. So that's the way we're interpreting it. But that's just speculation.
Great, thank you.
Okay, Adrian, you had your hand up. Would you like to ask your question?
Great talk as ever. You achieved over 700 attendees today, which I think is our record. Perhaps closer is the record, so brilliant. One question related again to the RNA interaction with PRC2, because at least my understanding is that there was the hot air model originally that long non-coding RNAs would directly bind to PRC2 and bring it in. And then more recently, people are thinking that that's not the case. PRC2 binds RNA generally or non-specifically. So I'm wondering, what is your view on this? Is it that some RNAs are bringing in PRC2 and some RNAs are pushing it away? Or because you mentioned that the RNAs you were looking at were perhaps encouraging PRC2 to put K27ME3 there. What were those RNAs and where do you stand?
I actually wasn't suggesting that, but anyway, let me just show you unpublished result here, that where we basically done RT and TAG using for H3 K27 trimethylation, which is what we know that EXIST will bring this in. So EXIST actually is a long non-coding RNA that'll bring it in. And we were intrigued. So first of all, you see that TSIX and EXIST both come up. Well, that's expected because they overlap at the 3' end. So they're basically picking up the same thing here. But what about all these others? Are these just nascent RNAs or are they actually doing something? But it turns out that if you look at the size, these are enormous. These are the largest transcripts in the cells. So what we're actually, whereas EXIST and TSIX are sort of normal RNA size. So what we're picking up here is basically we're only picking up the background of nascent transcripts, presumably because you're getting because there's poly A, there's a little bit of poly A that's getting there. But this also kind of says that at least for polycomb silencing, there's only EXIST in TSIX that it can be associated with this. So I think this is kind of a way that we can test these models that people come up with for hot air and all these others, where they're actually bound to the RNA. They might be doing something in the cytoplasm, we don't know. They might be doing something in nucleoplasm, we don't know. But we can actually test whether they're associated with the chromatin. This is something we're doing right now.
So I have a quick question there if no one else is jumping in. Sorry to go back to a technical one after a very interesting answer to Adrian's one. I was very impressed with you had slides ready for him already. You think very quick. So we're starting CUT&Tag in the lab, and I see you used normalization or some sort of spike in for your BRG1, either CUT&Run or CUT&Tag. So what's your opinion on the best way to normalize CUT&Run and CUT&Tag?
Well, we just do a global calibration using, you can add spike-ins. You can actually buy some spike-ins of nucleosomes. That's the best way to go. But you don't necessarily need that. If you use the homemade, we use our own homemade stuff, and we don't do anything to remove the excess E. coli DNA that comes in there, but that gives us the perfect spike-in in order to do the global calibration. I don't call it normalization because we're not actually making any changes. We're just doing to compare two samples that are treated with the same antibody. You can compare them doing that. But like I say, we're not normalizing, we're not dividing by input or anything like that. So there's no normalization, just calibration, that's good enough. And there are various ways that people have done spike-ins, and they seem to work pretty well.
Brilliant. Okay, Steve, I sort of thought we should have one last question. There's one question with 8 upvotes in the Q&A column there from Dan Coleman. If you'd like to answer that one, I figured.
So does the assay still work well? You're talking about CUT&Run. It's in physiological conditions. It's still in an intact nucleus, and so the trick here is just to make sure that there are no active processes going on. And the way we deal with that is when we add the cells might be live, or they might but they're unfixed. The trick is something we've been doing right from the start, is add a little bit of EDTA in the initial buffer that we're adding, in the antibody buffer. And that will stop all active processes because it sequesters magnesium, which is needed for ATP-dependent processes. So stop all remodeling, all polymerases, and that seems to work just fine.
Right. Stephen, I have just one burning question regarding open and closed regions for researchers interested on proteins on heterochromatic regions. Do you think CUT&Tag or CUT&Run have a bias, given the enzymes need to have access to the chromatin to do the cutting? So is there any bias towards you or heterochromatin regions?
Not that we can detect, and that's because, and I think the answer to that is we're tethering. Now, if you're doing something like accessibility mapping, of course, that's going to be a problem because the enzyme, whether it be eminase and we've done lots with eminase and see exactly what you're saying. But when it's tethered, whether it's eminase or TN5, when it's tethered, it's basically going to be only, that's really going to dominate over any other bias. So our favorite control is the one I showed you, H3K27 trimethylation, and that's of course compacted. Some would call it heterochromatin. And the other point is that for CUT&Tag, there's also the advantage that it's one and done. In other words, once it tagments, it's out of the picture. You can't do any more. So it goes to completion. And because it goes to completion, and because it's not going to be bothered, it'll even tagment over a nucleosome because it's really a powerful thing. Whereas eminase will just keep going. It's an enzyme, so you got to control it a little bit. So We do things at low temperature in order to control it. Does that answer your question?
Yeah, perfect. Thank you.
Okay, well, so since we've gone past the 5 o'clock hour here in Ireland, and that's past 6 o'clock in Europe, I guess we should draw to a close. I thought the most upvoted point that's left is actually a comment from Claudia Sanchez, who says, "Thank you very much for the protocols.ie resource." And I think that's a really wonderful comment on this whole technique, that it's so open and so accessible for people to use. So genuinely, thanks for that, Stephen. Thanks for the excellent talk as well. And we hope to see everybody. Well, firstly, Leo put a comment up that you can watch the Active Motif social media if you want to go back and watch the recording of this. And we hope we see you in a couple of months' time for Carolyn Luger to talk about chromatin structure at a more molecular level. Okay, so I think that's the end. Leo, do you have anything you want to say?
No, that's all.
Oh, I just want to thank people for the questions. They were great.
Thank you, Steve.
Brilliant. Thank you very much again. That was wonderful.
Thank you, Steve.
Bye.
Thanks a million.